Ziehl Neelsen staining or acid-fast staining, which is mostly done for the mycobacterium species, because this acid-fast staining is done for the organisms which are having mycolic acid in their cell wall. So, the specimen which we take for the staining is sputum, and the requirements for the acid-fast staining is strong carbonyl fission and 20% H2SO4, methylene blue, distilled water and liquid paraffin. So now the composition of strong carbonyl fission is basic fission of 10 grams, absolute alcohol of 100 grams and 5% phenol and water of 1000 ml. Now, opposite to the gram staining, where we'll be using the primary staining, we'll be using the methylene blue. And for the counter staining, which is secondary staining, we'll be using the strong carbyl fission. No, not strong, but dilute carbyl fission. But for the acid-fast staining, we'll be using the strong carbyl fission for primary staining and also the mordanting. For the gram staining, we'll be having 4 steps. But for the acid-fast staining, we'll be having only 3 steps, where the mordanting and the primary staining will be considered as 1 step. So, coming to the primary staining and mordanting. So, the primary staining, the smear is taken and the strong carbyl fission is added and heated for 5 minutes and heated on the non-luminous flame. The steam should come out, but the stain which we added on the slide should not boil. Only the steam or the smoke should come out. And washed with the distilled water. After 5 minutes of heating, it should be washed with the distilled water. Now here, the strong carbyl fission act as basic stain and the carbolic acid act as mordant, which is none other than fixator. Fixation. It as the basic stain stays on the bacteria. Now the decolorization. For decolorization, we'll be using 20% H2SO4. The washed smear is coated with 20% H2SO4 for 2 to 3 minutes and then washed. So this process will be repeated until the slide becomes colorless. Colorless mean bacteria which take up the pink color of strong carbyl fission will be remain same, but the non-acid fast bacteria should leave its color. So we will be repeating this step until the bacteria leave its color and slide becomes colorless. So here in the primary staining, what happens is the bacteria will be having the mycolic acid in their cell wall. So when we heated this, the mycolic acid which is present on the cell wall of the bacteria will get liquefied. And it allows the stain to get into the cells. So it acquires the pink color of strong carbyl fission. And now after decolorization, we'll be coming into the secondary staining, which is counter staining. So now we'll be using methylene blue for this for 1 to 2 minutes. Now again washed with distilled water and then blotted with the paper until it dried. So after this process completed, we'll be keeping cedarwood oil or liquid paraffin on it and focusing. We'll be seeing acid-fast organisms in pink color and non-acid-fast organisms in blue color. Now, acid-fast staining is mostly done for mycobacterium species like mycobacterium tuberculosis and lepore, and species like nocardia also will be using acid-fast bacilli. Now, the acid-fast seen in the bacteria having high content of mycolic acid in the cells. So, the bacteria which will be having a high content of mycolic acid only give positive results for the acid-fast. The bacteria which does not contain mycolic acid or will not have cell integrity, they won't give positive for acid-fast staining. So now this is the complete description of acid-fast staining. Thank you.